mutation rate, and then go on to what we actually found in that context. So what we did was we looked at ostensibly healthy families in Pennsylvania, in central Pennsylvania, and we wanted to have a high resolution view, but we needed to look at transmissions from mothers to offspring. If anyone doesn't know much about mitochondrial biology, mitochondria pass from mothers to offspring and not from fathers to offspring. So we looked at pairs of individuals, mothers and their children, male and female children. In follow-on studies, we've been looking at grandmothers, mothers and their children, to see more transmissions occurring. But what we did, which was a key thing at this early point in our study design, was to look not just at one sample from an individual, but two samples from different germ layers during development. So we looked at blood and we looked at the buccal cavity. So what we were examining here was, could we detect heteroplasmy, variants in mitochondrial DNA, present in not just one sample from an individual, but two samples that came from separate lineages early in development in that individual. And then do the same thing with the offspring, so that we were really looking at the possibility of transmission and also within individual transmission. And we looked at quite a large number of samples to do this too. Our basic approach was one where we had to be quite careful about falsely detecting, or false positives essentially. So to deal with this, we had a number of approaches. The first thing we did was to do what we call long-range PCR, to see if we could pull out mitochondrial DNA from samples where you wouldn't expect a large amount of it, such as blood samples. We weren't taking muscle biopsies or anything like that. So we took two long-range PCR reactions that overlapped and covered the entire human mitochondrial genome. The reason we were doing all this was primarily to avoid the problem, which is a well-known problem of pneumites, or nuclear copies of mitochondrial DNA. And pneumites are a huge problem for a study like this because during evolutionary time, there is essentially a shuffling back and forth between, or from the towards the nucleus from the mitochondria of genes, and that can result in you falsely seeing variation in the mitochondria when it's not actually there. The other problem is, of course, that some of these sequences that are present in the nucleus could even be polymorphic between individuals. So we really need a method that essentially excludes such sequences. If you just take blood, you take DNA from the blood, and you sequence it, you're going to potentially be making those sorts of mistakes, okay, where you're attributing variation to the mitochondria when actually it's just copies of mitochondrial DNA in the nuclear genome. So to deal with that, we took the first approach, which was long-range amplification, and the second approach, which was to use read lengths, so it's the fundamental unit of sequencing, that were very, very long, so that we would capture fragments that would include a full range of mitochondrial sequence, excluding those in the nucleus. And in reconstruction simulations, that was an appropriate approach. We also had to worry a great deal about contamination. Okay? So if you're working with the human samples, you should always worry about contamination. But also, if you're working with next-generation sequencing, there are a large number of reasons to be worried about contamination, particularly when you're asking questions about variation within a sample. You have to be very careful in that situation. And in fact, we did have a published study where we looked at all the variation that you could see in a sample, including a range of different strategies that we employed in this study to control for contamination. Computational methods we used were relatively straightforward. They involved conventional sequencing methods, conventional next-generation methods, followed by filtering to remove errors, followed by filtering to look very carefully at what we thought were variant sites, so regions that were varying between genome copies in an individual, and filtering them based on a number of known defects in sequencing. And then finally using some statistical approaches to try to estimate how confident we were that variation that we detected by sequencing was real. So we used a number of different methods to do that. And the fundamental unit of our analysis was a quartet, by which I mean a set of 4 samples, 2 from the mother, 2 from the offspring. And we would easily see patterns like this emerging, for example, where you have a mother, 2 samples carrying, say, 3% there of a mutant allele at that position in the mitochondrial genome, and the offspring would have ostensibly none, or below the detection threshold in our experiment. To be really sure that we were doing the right thing, we went a step further, and also when we saw a slide like this, we double-checked it. Okay, so what do I mean by double-checking? We looked using conventional sequencing of that specific region of the DNA, and we also used a relatively new method called digital droplet PCR, which is a particular variant of digital PCR that is becoming quite popular now among people who are trying to create more precise absolute quantitation of DNA. And then after all of this cross-validation and sequencing, we identified a range of different variants across the mitochondrial genome, with some not altogether surprising patterns. Certain regions of the mitochondrial genome are known to have very high rates of mutation, and the D loop, which is all the red circles on that diagram, is one such. I'm going to go a bit faster at this point, because there's a little bit too much data showed here. The key thing is that we saw mutations of different types in different places that were consistent with what we'd expect. And one of the key things that we found that we expected to see was that non-synonymous changes in protein-coding genes in mitochondria were depressed relative to expectations. So there was some evidence for maintenance of function in mitochondria in humans, in healthy humans. But also, we did actually find that about 1 in 5 of our families carried disease-associated mutations. So this might seem to be contradictory, but the key point here is that they're at very low frequency in general. So when a mutation is at a low frequency within an individual, it does not necessarily lead to disease. And this is one of the key reasons why it's interesting to study this variation. The other key finding that we had was to see that the relationship between levels of variation within individuals was generally higher or strong than it was between individuals. So fluctuations between generations were much stronger because of that germline bottleneck. It was that reduction in numbers of mitochondrial genomes that happens early in development after fertilization. Because of that, if you looked at Cortex and you looked at the mother samples versus the child samples, you'd see more variation in the frequency or the proportion of alleles in that instance than you would if you looked at, say, the child's 2 tissues or the mother's 2 tissues. And we were able, therefore, to categorize all the different types of variation into variation that we knew occurred in the germline, such as the second row there in the table, where the child has it and the mother doesn't, or variation that emerged de novo within an individual or was lost within an individual, which are what the last 2 rows indicate. If we looked in general at all the changes that we saw, we find that the fluctuation from one generation to the next was roughly consistent with most of the changes not being selected, but actually being fluctuating in a symmetrical and random way. So this is what you'd expect to see if genetic drift is the dominant force. If it's just a sampling process, a mutation either increases in frequency or it decreases in frequency. It has no particular tendency to do one or the other. And you can see roughly the same figure if you remove those non-synonymous changes or include them. And then the real point of all this is to try to measure what the size of that bottleneck is. Okay? Now we can't actually estimate exactly how many mitochondrial genomes there were, because mitochondrial genomes may travel in groups. For one thing, there's more than one mitochondrial genome per mitochondrion, but also it's possible that different types of mitochondrial genomes could travel together in groups. So we can't be 100% definitive and say the bottleneck is this size. But what we can say is that the effective bottleneck, a bottleneck, the perfect population that would produce the variation I just showed you, would be of about the size 35 in mitochondrial genomes. Okay, so we were able to estimate this by assuming that it was a binomial distribution and looking at the maternal minor allele frequency, so the rate at which you see the mutants, and then looking at the spread or the measure of variance that we had. To estimate measurement error, which is part of our measurement here, we did sequencing of clonal DNA, cloned mitochondrial DNA in the D-loop region, and we used that cloned DNA to estimate the rate at which our sequencing was defective and was giving us the wrong result. So in addition to all the controls we did to cross-validate, we did that to look at the possibility that our measurement of the actual level of mutation was also incorrect. Nevertheless, we did have quite a spread in possible sizes of bottleneck. But the other thing that this enables us to do is to start asking the question, what is the mutation rate? So essentially we're co-estimating these two properties, both the bottleneck size and then what is the actual rate at which new mutations arise. Okay, so I'm not going to completely justify that equation because I'll be honest with you, I can't quite remember all the details that went into that analytical result. But one of the key points to highlight from it is that we had to take into account the fact that we can't see all the heteroplasmids. We can't see all the variants in our samples. So if you can't see everything, that's a problem. And if you think about the variation, it could actually easily be that most of it is below the surface. You can't see most mutations because they flicker in and out of existence very quickly. But taking that into account and using our estimates of the bottleneck size, we were able to calculate a mutation rate that's about 10 to the minus 7 per generation or 10 to the minus 8 per year. And the D-loop region, as expected, has a higher rate of mutation. And the key thing is, what does that rate mean? What's the significance of it? Well, it's reasonably consistent with rates that were estimated from phylogenetic studies comparing species. So that's quite reassuring to us, but it's generally a little bit lower than most pedigree estimates. Okay, so that was also quite interesting to us as well, to see that it fit reasonably well within the literature and also fit quite well with phylogenetic studies. A potential interest in terms of understanding the risk of passing on mitochondrial illness is to see what the effective maternal age is on heteroplasmids. So the first thing to ask would be to say, if the mother is older, if she's an older individual, does she herself have more heteroplasmid? Is she more likely to have higher levels of heteroplasmid? And it's not a strong effect, but yes, there is an effect there, a significant effect, and an increase in the heteroplasmid level in older mothers. But also, of course, if the mother was older when she gave birth, or at conception rather than more relevant, then we see indeed an effect on heteroplasmids in the child too. So there is a maternal effect on the mitochondrial mutation rate as well. Or rather, not on the rate, but on the number. So we saw quite a severe reduction in numbers of mitochondrial genomes and an association between the maternal age and the number of heteroplasmids that we had. And a figure that was reasonably consistent with the literature, and also some evidence that many of us are actually carrying around heteroplasmids, me included, by the way. So. Okay, so I have a few minutes left to just talk about how I'm going to try and pull some of these. It's probably a bit quixotic this, but I'm going to try and pull together some of these different themes. So what I've really been talking to you about, some of the time I've been focusing on methods. How do we know whether there are a small number of mutants present in a large population? To all intents and purposes, an infinite population. And then what are the consequences of changing the rate at which mutants enter into the population, which is my current research topic of interest, which is lethal mutagenesis. But I also want to put mutation into a slightly broader framework, a framework looking at the possibility that an organism's variability itself is a property subject to selection. And one has to be very careful when you do this, when you talk about the possibility that something is selected indirectly. So the first thing is to say, so I'm talking about this concept of bet hedging, to hedge one's bet. And you should immediately be careful about everything I now say, because I'm using metaphorical language. In my first slide I said one of the big dangers, the second slide, one of the big dangers of doing evolutionary biology is inappropriate use of metaphorical language. So health warning with all that follows. The first thing is to say what is not bet hedging. And one of the things that's important to realise about a situation in which a population consists of competing individuals is that the fitness of individuals relative to each other is actually a diminishing function of their absolute fitness. So what do I mean by that? What I mean is that if an individual has a mutation that makes them more fit, that is actually arithmetically diluted a little bit relative to having a mutation that makes you less fit. So, in other words, you might expect evolution, to use slightly anthropomorphic language, to be conservative, to favour not losing fitness more than it favours gaining fitness. Okay, but that's not, for simple arithmetical reasons that I'm trying in a range of perhaps not successful ways to illustrate on the screen there, is that an extra unit of benefit is not as important as an extra unit of cost. That's not exactly what I'm talking about when I talk about the idea of bet hedging, the idea that variation is an important process. But what I'm talking about here is a different kind of conservatism. It's the fact that if fitness is something that varies randomly, it's not an absolute property. You don't look at someone's genome and then say you are this fit. If it's a random variable, then one of the things that might be favoured by selection is the consistency in your fitness. The simple way of thinking about this is that you have a lineage of organisms, and one individual in that chain, in that lineage, has a fitness of 0, and your lineage is now extinct. Okay? Very simple way of thinking about it. So a very high variance in fitness is not a good thing. So you would expect, and this is the essence of what I mean by bet hedging, or what others mean by bet hedging, is that you would expect there to be some degree to which the variance or variation in fitness is diminished, even if that itself lowers the fitness. So you might settle for a lower fitness, even if it causes, even if it has that cost, because of the benefit of reducing the variation. And that can happen in 2 ways. One is to give rise to conservative offspring, so offspring that have a rather boring phenotype perhaps, but a phenotype that's resilient and robust and is of reasonable fitness in all possible circumstances. The other is to produce a range of different types of offspring, some of which have a 0 fitness and some of which have a high fitness. And these are generally distinguished as quite different types of bet hedging, but in fact really analytical work suggests that they're just on a continuum with each other. And the key thing to understand is the relationship between variation in fitness within individuals and also within a lineage. The key point being that simple example I gave you, where one individual in a lineage has a fitness of 0. So I'm thinking about this from the perspective of viruses. Because viruses, if you like, are, you might think, why would viruses have this problem? You know, they surely have no problems at all. They just produce billions of offspring. But in fact, they do have the potential problem of not being able to understand what the environment holds for them. The variation in the environment is substantial for bacteriophage. Now, one of the most famous ways in which bacteriophage cope with a variable environment is through what we call lysogeny, which is the ability to switch between 2 different life cycles. One in which you survive in the bacterial host and you wait out the bad conditions. The other is in which the rats jump off the sinking ship when conditions get bad and start to replicate by the usual viral life cycle, just mass murder, essentially. And that switch between those 2 life cycles is an example of what we conventionally call regulation. In fact, it is the example I teach when I teach genetic regulation at undergraduate level, is the lambda switch, the ability to switch between those 2 life cycles. However, other possibilities have also been demonstrated in different viruses, where sometimes viruses will simply not infect their host. They will be a so-called residual fraction that sticks around and doesn't result in the death of the host. So I'm going to go quickly here because there is limited time, but just suffice it to say that testing for the existence of that is possible in the context of my virus. And one of the interesting ways to approach that, the last bullet point there, is by precisely controlling both the density and the generation time of the host. So if you change the length of time it takes for the host to replicate, or if you change the density of hosts so that the next host is either easier or harder to find, you can start changing the trade-off, the payoffs associated with moving from one virus to the next. So that's one approach to the problem. I'm skipping a little bit quickly there. And finally, it would be interesting to explore the intersection between these processes and the process itself of regulation. And you link these 2 together. And again, you have to be very, very careful about this. But can you understand the link between starting to take control and take a signal from the environment about expected variation versus simply being able to tolerate unexpected variation? So the sort of a very loose way of thinking about this, is nature neurotic, or is it busy planning to find out what's about to go on? And one of the approaches that we could take towards this is looking at looking at a simulation where we examine the relationship between the rate at which variation occurs and the reliability of getting a signal about what that variation is going to be. So looking at the borderline between reliable regulation and response to the environment versus not being able to predict sudden changes. And of course that's potentially of interest in general when you think about what it means to invest in reliably understanding your environment. How do you know what the significance of variation is in your environment for your fitness? So there could, of course, be a relationship there with the number of offspring that you produce in your fecundity. Anyway, I'm going to pop to the acknowledgements now and say thanks to the following folks. So thanks to my PhD student who's doing the work on the lethal mutagenesis. thanks to some MSc and Erasmus students I've had who have worked with lethal mutagens, and one of whom I'm interviewing tomorrow for a PhD position. Some of my colleagues at NTU who've been particularly helpful with methods, especially of late. Family members, including Tom, who didn't properly acknowledge on the last point, and former advisors of mine at Penn State University, with whom I worked on the mitochondrial project, and there is also a separate series of funding acknowledgements that are required for that. And of course, the very important people, the people at the Hershey Medical Center in Pennsylvania, actually collected the primary samples from patients, which I then went on and processed and did all the work on. So it's very important to acknowledge the people who actually did the sample collection. So, thank you very much.
Do we have any questions for Ben? Easy ones, hard ones, counter examples? Or maybe it's something we can take to the discussion when we have some coffee and some pastry. Oh no, we have one here, please.
Simple one. So for the mitochondrial study, you use long-range PCR, and then you sequence the products. PCR is prone to biases. Could that have affected some of the results? Would that be better to centrifuge first and just select the mitochondrial portion?
Yeah, I mean, we were a bit limited there by the kind of samples that we had for pre-selecting mitochondria. We, of course, tried at quite a lot of length to use different strategies. A rival research group had been doing 3 long-range PCRs of different sets of primers, and we tried that and had no observable differences. So it depends on what type of bias you mean, but it's something of constant concern to us whether we were reliably looking at DNA. Of course, we used and published information about known new mites to try and avoid those, but that's obviously not all on itself.
Can I ask a really broad question?
Go for it.
It's coming from a psychologist, so pardon my gross biology. It sounds like you talked a lot more about, I suppose, what in old terms would be called endosymbiotic prokaryote, so this kind of relationship between the host and the organism. Are the selection pressures different for free-living prokaryotes?
Yeah, that's a very important point, actually.
So you talked a bit about the host, and then you talked about checking out the host in the experiments, but is there something going on? Is there a difference in the evolution of small genomes, I suppose, between those two?
Yeah, I mean, there's quite a radical difference, even I mean, that's a classic area when you're talking about things like the C-value paradox and why genomes have the size they do, which is that there's known to be a reduction in genome size in intracellular parasites. I mean, the bottleneck itself, the existence of the bottleneck, is interesting, and there is evidence from work in mice that there's quite strong selection involved with the bottleneck, but also having that kind of process occurring where you end up with only a few types in the next generation could be of benefit. I mean, it becomes of interest about levels of selection. It's a benefit to the organism as a whole to have only one type of mitochondria present, perhaps. I have to be very careful what I say there. But yes, you would expect different pressures to operate.
What did you want to say?
What did I want to say? Well, yeah, I mean, there's also the very famous example of the petite mutation in yeast, which is a mitochondrial mutation that results in enhanced replication of smaller mitochondrial genomes. So one of the things you could go looking for is the relative difference in transmission according to selection at those different levels on mitochondria themselves directly, and also of the design of the inherited system, if you like, which seems to reflect the fitness of the individual. So that's an area where I feel slightly underconfident because I have to sit down and think about what the time constants would be on those different processes, and I'm sure other people have done that as well. So.
Any further questions? We can certainly take our questions to the coffee and cake, but if we thank Ben once more